Welcome to nitric oxide and amines. Let us understand what are these biogenic amines. Biogenic amines are formed when an amino acid undergoes decarboxylation. It forms the respective amine releasing carbon dioxide. Okay, examples include histidine. Histidine undergoes decarboxylation to form histamine, and this histamine is responsible for all allergic reactions and anaphylaxis. Tyrosine undergoes decarboxylation to form tyramine, and this excess tyramine stimulates release of norepinephrine, which acts as vasoconstrictor. Tyrosine tyrosine gets converted to dopa by tyrosinase enzyme, and dopa undergoes decarboxylation to form dopamine, and this dopamine acts as a neurotransmitter and it is also responsible for memory. Tryptophan undergoes decarboxylation to form tryptamine, otherwise known as serotonin, and this serotonin is responsible for regulating the mood, appetite, and temperature. Apart from that, serotonin also induces sleep, reduces pain, and it acts as antidepressant. Glutamic acid undergo decarboxylation to form gamma-aminobutyric acid, and this acts as inhibitor neurotransmitter in our central nervous system. Lysine undergoes decarboxylation to form cadaverine, and this cadaverine is responsible for cell growth and proliferation. Another significance of lysine is that, especially the cadaverine, is that it accumulates in the decaying tissue and it is also used as marker of putrefaction. The last one is ornithine undergoes decarboxylation to form putrescine. Putrescine undergoes decarboxylation to form spermidine, and spermidine undergoes decarboxylation to form spermine. So this putrescine, spermidine and spermine are polyamines. So the question that is usually asked with respect to this is enumerate different biogenic amines and mention their function. This may be asked as a 5-marker question. Coming to polyamines, now the most probable question that is asked with respect to polyamines is what are polyamines and what are their functions. Now polyamines are polycations that are responsible for cell growth and differentiation. There are 3 polyamines: putrescine, spermidine and spermine. Why they are called as polyamines? Because they contain multiple amino group. Now these are biosynthesized in our body from methionine. Methionine undergoes activation to form S-adenosyl methionine, wherein the adenosyl group is added to methionine and the adenosyl group donor is ATP. From this active methionine, that is S-adenosyl methionine, carboxyl group is removed and it gets converted to decarboxy S-adenosyl methionine. So this combines with ornithine. Now ornithine comes from urea cycle. So wherein carbon dioxide condenses with ammonia to form carbamyl phosphate, which reacts with ornithine to form citrulline. So this in turn reacts with aspartate to form arginosuccinate, and this undergoes lyase reaction to form arginine and ornithine, and this ornithine reacts with decarboxy S-adenosyl methionine to form putrescine, and the enzyme is ornithine decarboxylase. Now putrescine is the primary polyamine. This in turn reacts with DSAM, decarboxy S-adenosyl methionine, undergoes decarboxylation to form spermidine, which is also a polyamine. This is a secondary polyamine. So this further condenses with decarboxy S-adenosyl methionine, undergoes decarboxylation to form spermine. So this putrescine, spermidine and spermine, all 3 are polyamines. Now why do we need polyamines in our body? They are required for protein synthesis, especially they stimulate the initiation factor during translation. Apart from that, these polyamines are responsible for cell proliferation, stabilization of ribosomes. They are required for DNA and RNA synthesis and also protects the DNA against depurination. So one important clinical significance associated with polyamines is they are increased in cancers, all type of cancers. Now, so one more clinical significance associated with polyamine is this ornithine decarboxylase enzyme is inhibited by a drug called difluoromethyl ornithine. Now, this drug is used in the treatment of sleeping sickness. Now, the sleeping sickness is caused by a protozoa, especially trypanosomes. Now, so this DFMO, difluoromethyl ornithine, will inhibit the ornithine decarboxylase enzyme in trypanosomas. So what happens is there is no synthesis of polyamines, no cell proliferation, and this results in death of the trypanosoma. So this leading to cure of sleeping sickness. Now, so humans are not affected when this drug is given because the half-life of ornithine decarboxylase is in minutes when compared to half-life of trypanosoma. Half-life of ODC in trypanosoma is many hours. So it is synthesized very rapidly in humans, so it has got no effect on the human cells when compared to trypanosoma, where the half-life is many hours. So there occurs inhibition of synthesis of polyamines. So no, there is no cell proliferation in this parasite, resulting in cure for sleeping sickness caused by trypanosoma. Now coming to nitric oxide. Now this can also be asked as a 5-marker question for you. Now this nitric oxide is uncharged unpaired electron, so it's a free radical. Now it is also synthesized in the body from the amino acid arginine. Now arginine is involved in the synthesis of nitric oxide and catalyzed by the enzyme nitric oxide synthase. Now this is a multi-enzyme complex. It requires many coenzymes for conversion of arginine to nitric oxide and citrulline. Now the coenzymes, it requires molecular oxygen which gets reduced to water, and the hydrogen atoms are donated by tetrahydrofolate which gets converted to dihydrofolate. So the reduced form is generated by heme which gets oxidized to ferric form. This is regenerated, the ferrous ion is regenerated with the help of hydrogen atoms donated by FMNH2, active form of riboflavin, which gets oxidized to FMN. So the reduced form is regenerated by FADH2, again the active form of riboflavin, and this gets oxidized to FAD, and the reduced form is regenerated back by NADH, the active form of niacin. So all these coenzymes convert arginine to nitric oxide and citrulline, catalyzed by the enzyme nitric oxide synthase. Now this is isoenzyme. It is present in 3 different tissues. The neuronal nitric oxide synthase is there, endothelial is there, and the macrophage is there, which have got specific functions in our body. The neuronal nitric oxide acts as neurotransmitter in the central nervous system, responsible for release of corticotropin releasing hormone and gonadotropin releasing hormone from hypothalamus. It increases GIT motility, also responsible for retrograde memory. Endothelial nitric oxide is responsible for dilatation of the blood vessels and inhibition of platelet aggregation, whereas the macrophagic nitric oxide protects the white blood cells from and also the body from attack of microorganisms. Now what is the metabolic fate of this nitric oxide? The half-life, just check out, it is hardly 3 to 4 seconds. Now nitric oxide combines with oxygen to form nitrates and gets excreted in urine. It can combine with hemoglobin to form nitrites, and this also gets excreted in urine. And nitric oxide as such can be expelled from the lungs during expiration. It can combine with superoxide anion to form peroxynitrate, and this can cause damage to the lipids, resulting in lipid peroxidation. So clinical significance, it has got, nitric oxide has got a great clinical significance, especially the endothelial nitric oxide, which is responsible for blood vessel dilatation. Now this property has been used in the treatment of angina pectoris. Angina pectoris is the chest pain caused by spasm of the coronary blood vessels or narrowing of the coronary blood vessels due to atherosclerosis. Apart from that, this property is also utilized in the treatment of pulmonary hypertension, where the blood vessels supplying the lungs are narrowed because of increased pressure. So in both this condition, so endothelial nitric oxide is used as therapeutic agent. So there is one more significance associated with nitric oxide is there is increased production of nitric oxide is sepsis. In sepsis, sepsis is a dangerous condition where there occurs multi-organ failure because of microorganism invasion, and increased production of nitric oxide in sepsis. What happens is there is massive dilatation of the blood vessels, resulting in profound fall in blood pressure, and one worst thing is this fall in blood pressure does not respond to any vasoconstrictor. So these are the clinical significance associated with nitric oxide. The probable question that is asked is, how is nitric oxide formed in the body? So you need to remember, one line answer is it is formed from arginine catalyzed by nitric oxide synthase enzyme. So it is better you write this part of the flow chart. Now the other question that can be asked that is coupled with the first question is, what are the functions of nitric oxide? So you need to write specific functions for all the 3 isoenzymes of nitric oxide synthase. Neuronal nitric oxide responsible for being a neurotransmitter, release of gonadotropin releasing hormone and corticotropin releasing hormone from hypothalamus, increases GIT motility, responsible for retrograde memory. Whereas endothelial nitric oxide dilates the blood vessels and inhibit platelet aggregation, and macrophagic nitric oxide provide protection against microorganism. So this was all about polyamines nitric oxide. Time for assessment. Which of the following is a polyamine involved in cell growth and proliferation? Answer is putrescine, which is a primary polyamine. Polyamines are synthesized from which amino acid? Answer is ornithine. Ornithine comes from cycle. The enzyme responsible for conversion of ornithine to putrescine is ornithine decarboxylase. Spermidine and spermine are important for, answer is DNA stabilization and cell proliferation. Increased polyamine synthesis is commonly seen in cancer cell, but it is a non-specific. Which of the following is derived from histidine? Histamine is derived from histidine by decarboxylation, and this histamine is responsible for mediating anaphylaxis and allergic reactions. Dopamine is synthesized from tyrosine. Tyrosine gets converted to dopa. Dopa undergoes decarboxylation to form dopamine, which acts as neurotransmitter ganglia. Biogenic amines are mainly inactivated by monoamine oxidase, which converts the amines to safer aldehydes. The cheese reaction is caused by accumulation of tyramine. Tyramine is formed from tyrosine. Now this tyramine is degraded by monoamine oxidase. In patients taking monoamine oxidase inhibitor, tyramine is not getting degraded. It gets converted to norepinephrine, and this norepinephrine produces profound vasoconstriction, which is known as cheese reaction or the hypertensive. Nitric oxide is synthesized from arginine by nitric oxide synthase. The enzyme responsible for nitric oxide synthesis is nitric oxide synthase enzyme. So nitric oxide acts primarily as neurotransmitter and vasodilator. Now this vasodilator property is used in the treatment of angina pectoris and pulmonary hypertension. So which drugs works by enhancing nitric oxide mediated cyclic GMP pathway? Sildenafil. Now this has been used in the treatment of erectile dysfunction, where it acts as vasodilator. So thank you. So this was all about polyamines and nitric oxide. So to have a comprehensive approach, watch all the videos on protein metabolism which have been placed in the playlist and named as protein metabolism for having complete clarity. So next video will be on inborn error of metabolism and their screening test. So if you are like this video, please do like, share and subscribe. Thank you.